Hello from ChemHelp ASAP. Let's discuss how impurities in drugs can lead to safety risks for patients.
Upon completion of this video, students will be able to list sources of chemical impurities, quantify impurity levels that require action by the discovery group, and describe how impurities affect how drugs are produced and tested.
Small molecule drugs, drug candidates, and clinical candidates are chemicals. While the compounds are ideally pure, 100% pure, no chemical is 100% pure. Everything has impurities. What is the origin of impurities in a research compound like a drug candidate? Small molecules are synthesized in a lab, and they may contain small amounts of unreacted reagents or starting materials. Chemical reactions can produce side products or isomers, especially stereoisomers and regioisomers that can be difficult to remove through purification. Solvents from reactions can remain trapped within the crystal lattice of a solid product. Metal complexes used to catalyze reactions are often notoriously difficult to remove. Finally, impurities can also arise from degradation of a molecule that has been stored too long or under improper conditions. These impurities can have their own biological activity and can lead to additional safety risks. Different threshold levels of impurities trigger different types of action by regulatory authorities. The actions depend not only on the percentage of the impurity, but also the size of the daily dose of drug. So the action required varies based on the amount of impurity that a patient receives. There are three types of actions. The first level is reporting of the impurity. For the lower dose category, any impurity at 0.05% or higher must be reported. If the impurity is 0.10%, then the impurity must be identified, meaning the structure of the impurity must be determined. Simply identifying the impurity is an adequate action as long as the structure of the impurity is not suspected of causing toxicity. Once the impurity reaches 0.15%, the impurity must be qualified. So the impurity must be identified, separately synthesized, and tested for toxicity. For higher dose drugs, the thresholds are set to lower percentages. So as an example, the maximum daily dose for atorvastatin, a common cholesterol-lowering drug, is 80 milligrams per day. The 0.05% reporting threshold is just 0.04 milligrams. The 0.1% reporting threshold is just 0.08 milligrams. The 0.15 qualification threshold is just 0.12 milligrams. The threshold for some metals, such as palladium and platinum, which are fairly common catalysts, are only allowed down to 10 ppm. That's 0.001% for oral drugs. The cutoff is even lower, just 1 ppm for injected drugs. The need to minimize impurities creates several challenges. First, the production method must be very reproducible in order to maintain consistency from one batch of the drug to the next. Second, the method of analysis must also be consistent and validated to be able to detect and quantify small impurities. Third, the clinical trial material should be ideally at least as pure as the material used for preclinical toxicology studies. If the clinical trial material is less pure, the dose limits will be reduced because of the higher impurity levels. Finally, the detection of new impurities in a batch is very problematic because the safety risks of the new impurities will not have been studied. In extreme cases, new impurities can result in the loss of an entire batch of drug.
We listed origins of impurities in tested compounds. We have quantified impurity levels that require action by the discovery scientists. Finally, we have described some ways in which the need for high compound purity affects the late discovery process.
I hope you enjoyed this video and found it informative. Your feedback is always welcomed and read. Please leave any comments in the section below the video. Take care.